The ST-deconvolve accurately recovers cell type proportions in multicellular spatial transcriptomic tissue profiling. Miller et al. Present a visualization where the components of the deconvolved cell type pixels are illustrated as pie charts, a standard visualization for proportional relations. They can be superposed to the respective hematoxylin and eosin, H and E, stain, as the locations of pixels is given by the center of each circle. The visualization has to convey two vital types of information: locations and proportions. One of the fundamental tasks of visual analytics is to recognize coherent areas of homogeneous cell type compositions. A quest that is inherently difficult by comparing pie charts, as quantities are not aligned across pie charts. Besides pie charts, other well-known designs for visualizing part-to-whole relationships are bar charts, stacked bar, column charts, or tree maps. While suitable for the comparison of quantities between locations, bar charts come with the obvious downside that they exhibit a suboptimal shape for conveying locality. It is difficult for humans to perceive a chart's center. Also, their layout poorly uses the available space and does not scale well for a larger number of components. Stacked bar charts can be realized with a quadratic design. Same as with pie charts, we observe a level of spatial disarray of quantities, making it hard for viewers to spot homogeneous regions. Also, in regions with multiple equally strong quantities, it is comparably difficult to decipher their respective contributions. Tree maps make optimal use of the available space, which is iteratively split into regions reflecting the quantities' respective contributions. It is a very helpful design for a single ST pixel, but using tree maps in a small multiples visualization leads to a very turbulent-looking result. In summary, none of the baseline visualizations for proportional relations leads to a substantial improvement for the problem at hand. More advanced approaches for displaying small multiples in their spatial context exist, but are not directly applicable for showing proportional relations. We propose a novel design which optimally uses the available space, being as translucent as possible, while still conveying the proportional information. Markers should convey both proportional information and locality information. Additionally, we want them to visually reflect their association to local clusters. The core idea of our proposed design is to visualize the different cell type contributions as separated, yet tightly packed, smooth shapes, which reflect both a component's contribution and its belonging to local accumulations. We equip a circular base shape, reflecting the magnitude of the contribution, with a protruding spike, pointing towards the center of such a local cluster. The extent of this spike reflects the size and proximity of the respective cluster. This marker design emphasizes latent borders of regions with similar cell type contributions and facilitates the recognition of coherent regions. Regarding the positioning of the individual components within a ST pixel, we intend to arrange them such that they appear to be drawn towards their belonging local cluster centers. This ensures both that components' markers are closer to their respective clusters and that their tails are unlikely to obfuscate other markers, while still maintaining a tight packing. We achieve this layout through a physical force model with three forces simultaneously acting upon each contribution. A gravitational force, pulling them towards the pixel's center. A fast decaying collision force, preventing them from obfuscating each other's, and a clustering force, pulling them towards the local cluster center. In most cases, this leads to a blossom-like arrangement with differently large and elongated petals, facing away from the pixel center. We qualitatively evaluate our design by visually comparing it to the original pie chart layout using the given H&E image. Already from the macro perspective, it is apparent that our proposed design reveals several fine-grained intensity structures which are latent in the pie chart's layout, such as the turquoise X5 cell types, present all around the border, or the violet and pink marblings of the X8, X9 cell types to the top and left borders of the tissue sample. A closer inspection of the regions where homogeneous cell type compositions border each other clearly demonstrates the advantage of our design. This part of the tissue exhibits a coherent region, dominated by the red X1 cell type, which encases a cluster of the green X4 cell type. To the top and right we observe an area dominated by the X5 cell type which is intersected with a streak, composed in equal parts of X3 and X9 cell type. This also correlates with darker marbling in the H&E image. Even though this information can also be derived from the original pie chart visualization, our proposed design conveys this information more efficiently with less visual disturbance. Another example are regions where a dominant cluster has offshoots into neighboring regions. For example, the X1 cell type cluster in the center of the tissue. Here, the droplets shape leads to a more pronounced cluster outline, in accordance with the common faith Gestalt principle. For a proof of concept, visualizations were generated with a Python script without optimization in mind. The implementation runs fast on commodity hardware but will need further improvements regarding efficiency if the concept is to be applied in real time on tissue data. To this end, many approaches such as parallelization or approximation are possible and will be part of our future work. To extend the outreach of our droplets design, we also plan to produce a publicly available JavaScript implementation. Besides improvements regarding the implementation, we plan to subject our design to a rigorous perception-centered evaluation with users. Such will incorporate different visual analytics tasks which have to be performed using droplets as well as comparable designs. To summarize, we propose a novel visualization concept for illustrating the proportional relations in ST pixels of H&E images. Our design with droplet markers for indicating cell type contributions enhances a viewer's perception of coherent regions and provides an overall less turbulent overlay than superimposed pie charts. Efficiency and interactivity was no consideration during our experiments, but will be the target of follow-up work.